DNA resides in the nucleus of almost every human cell and contains the blueprints and information necessary for our bodies to work. Proteins, which are needed for function, structure, and regulation of the body, are made by a process called protein synthesis. It is a two-part process. First, the information in nucleotides from DNA are copied into RNA inside the nucleus by the process transcription. The RNA, which is much smaller than DNA, is able to leave the nucleus into the cytoplasm. Then, during the second step, translation, mRNA, tRNA, and a ribosome synthesize a protein in the cytoplasm. Now we will go in depth into each of these processes. The protein synthesis process begins in the nucleus with transcription. The first step is initiation. The two-stranded double helix of DNA is unwound and separated at the beginning of the gene sequence. RNA polymerase, the enzyme vital for this process, binds to a promoter, or initiation site, at the beginning of the gene sequence. The RNA polymerase enzyme moves along the DNA template strand until it hits a start codon, AUG, beginning the second stage, elongation. DNA bases A, T, C, and G are synthesized by the enzyme in triplets, forming complementary pairs on the RNA strand, just like that. As the transcribing polymerase moves along the DNA strand, the mRNA peels away. Termination ends the process when the polymerase reaches a DNA sequence with a termination signal. The RNA then detaches and is now messenger RNA, ready to move on out of the nucleus. Now, transcription is a very fragile process, and things can go wrong during the coding of ATCG bases and their pairs. When genes are read or transcribed incorrectly, this is called a mutation. They can either be benign or very disastrous to protein synthesis.
Splicing is the process of removing non-coding sections from the pre-mRNA strand. It occurs to the pre-mRNA prior to translation. mRNA is made up of exons, protein-coding sections, and introns, non-coding sections. In order for the mRNA to give the proper message, the introns must be spliced or cut out of the section. This is done by spliceosomes, which are composed of SNRPs or ribonucleoproteins, a mixture of RNA and proteins. The spliceosomes cut out the introns from the mRNA and pinch together the neighboring exons. The strand without any introns is now mature mRNA and can express the final set of instructions for the protein. Translation is the final step in protein synthesis.
This is where a ribosome is made up of large and small subunits that attach to the messenger RNA. Ribosomal mRNA is made in the nucleolus, and mRNA is made in the nucleus, which enters the cytoplasm through the nucleopores. The messenger RNA is read 3 bases at a time. Each sequence of 3 is called a codon and is made up of the base pairs A, G, U, and C. The transfer RNA delivers the corresponding amino acid. Each tRNA has 3 base amino acids, which are complementary to the mRNA's codons. Complementary pairing is when 3 base pairs match up to the RNA's anticodons. The anticodons are the base pairs on the tRNA. The catalytic site is where the amino acid chain is formed in the large ribosomal subunit. The A site is the acceptor site. P site holds the tRNA, which is where the growing polypeptide chain is, and the E site is the exit site. The chain is complete and is signaled to stop by reading a stop codon. The amino acid chain elongates and folds into a protein as it is formed.